Hello, my name is Jill Corre. I work in the unit for genomics in myeloma in University Cancer Institute of Toulouse in France. And I'm here to talk about an IFM proposal for risk scoring in multiple myeloma. Among all prognostic factors described in myeloma, the chromosomal abnormality have a deep impact. And those on which there is a general consensus and well established are the deletion 17p, 8% of patients, and the T414, 12 to 15% of the patients, which affect both PFS and OS. The question is, can we be satisfied with this dual deletion 17p and T414 to define today's myeloma high-risk patients, in particular with the perspective of risk-adapted strategy? The answer is clearly no. First argument, the heterogeneity of high-risk patients. For instance, we all know that there are good T414 and bad T414. And one explanation, among others, may be the presence of concomitant prognostic factors, such as here deletion 1p32. Patients displaying both abnormalities have clearly not the same prognosis than patients with T414 alone. Another important issue is the role of concomitant trisomies, and especially of which trisomy are we talking about. Trisomy 3 and 5 have been shown to abrogate T414. Look at the green curve, T414 with no trisomy. The yellow curve, T414 plus trisomy 3 or 5, which is equivalent to the blue curve, T414, no T414 and no trisomy, the best being the red curve, no T414 and trisomy 3 and 5. These trisomies abrogate T414, but only improve deletion 17p. The yellow curve, deletion 17p plus trisomy 3 or 5 is better than the green one, deletion 17p with no trisomy, but less good than the blue curve, no deletion 17p and no trisomy. In contrast, the trisomy 21 has been shown to worsen the prognosis of patients with T414 and deletion 17p. It's not shown here. Second argument, other prognostic factors have been identified to define high-risk multiple myeloma. 1q gain, and more recently, deletion 1p32. And third argument, the beneficial impact of proteasome inhibitors and immunomodulators for some high-risk patients. To try to better define cytogenetic risk, we simultaneously analyze all recurrent chromosomal abnormalities in a large cohort of newly diagnosed multiple myeloma patients included in the IFM trials. Our main objective was to develop a prognostic model that can be used routinely by our colleagues clinicians. We used 4 cohorts, 1 for training and 3 for validation, with a long follow-up and different treatment. This is very difficult to find a good compromise between a sufficiently long follow-up and a sufficiently updated treatment. The final model retains 6 abnormalities as statistically relevant. 5 associated with a poor prognosis, T414, deletion 17p, trisomy 21, 1q gain, and deletion 1p32, and only 1 abnormality associated with better prognosis, the trisomy 5. We then define a prognostic score where each abnormality is associated with a specific weighted prognostic value. Our prognostic score is a sum of the weighted values of the present abnormalities among the 6. Here you have the distribution of our score in a newly diagnosed population. This is a linear predictor of risk, with the best score being minus 0.3 and the worst being 3.2. And based on the 5 years survival probabilities, we also created 3 categories of risk. The low risk included all patients with a score less or equal to 0. The medium score included all patients with a score of 0 to 1, and the high risk group, high risk score included all patients with a score greater than 1. And here you have the overall survival according to these 3 categories. And here you are the validation of the 3 categories. And even in the external validation dataset 2, whose probability whose survival is better thanks to an updated treatment, our score retains good discriminatory abilities, suggesting that it may contain the main cytogenetic lesion. Here is the association between the 6 abnormalities in the 4 cohorts. 20 to 25% of patients have no abnormalities. The median number is 1 to 2 abnormalities per patient, and the frequent combinations were trisomies between themselves and the presence of 1q gain in patients with T414 and in patients with a deletion 1p32. According to our score, the distribution of risk in newly diagnosed myeloma was the following. 55% of patients are in the low risk group, 35% of patients are in the medium risk group, and the remaining 15% are in the high risk group. Importantly, 94% of patients with deletion 17p were high risk, but only 29% of patients with T414 were high risk. Using external validation set 1 and set 2, we also test the discriminatory ability of our score when compared to ISS stage or to the duo T414 or deletion plus deletion 17p. And we showed that our score had the better discriminatory ability, either used as a linear predictor, the model 1, either used with the 3 risk categories. When we combined our score to ISS, the added value was not clinically relevant. And finally, when we compare to revised ISS, thanks to the external validation set 2, we showed that our score have a better discriminatory ability than the revised ISS. One limitation of our study is the non-inclusion of T1416, which is included in the revised ISS, along with deletion 17p and T414. This translocation being rare, only 3% of patients, its independent prognostic value is difficult to establish, and data are still not clear today. A very important issue is the clonal size of deletion 70p to have the prognostic value. The IFM published more than 10 years a study showing that this cutoff was 60%, and we use this 60% cutoff to develop our prognostic score. But no other group did address this issue, and this leads to very confusing data about the impact of novel therapy for abrogate deletion 70p prognostic value, with some investigators defining deletion 17p when present only in 1% of plasma cells. We recently addressed this issue by participating to a meta-analysis with more than 1,000 patients with deletion 17p at all levels, and we confirmed the IFM data with a cutoff of 55% to have the better prognostic value of deletion 17p. And finally, double-hit multiple myeloma have emerged as a new high-risk myeloma group, raising two important issues. First, the question of the number of copies of 1q. Do we need to have an amplification, meaning at least 4 copies of 1q to have the prognostic value? And the second one is the question of TP53 mutation. Do we need to have a double hit on deletion on 17p to have the prognostic value? In conclusions, only deletion 17p can alone confer a high risk by itself. T414 should not longer be considered as a high-risk factor by itself, but should be interpreted in the light of other abnormalities around it. We can't define high-risk myeloma with 2 or 3 isolated abnormalities. This classification is oversimplified and leads to a misclassification. The score presented here is just a score, far from perfect, but it's innovative because it includes several cytogenetic abnormalities which are weighted, and it's also innovative because it includes protective abnormality. And this score is very easy to use. It can be used routinely, even in labs where the only available technique is FISH. There is an unavoidable dissociation between the design of clinical trials and the evolving definition of high-risk patients because of new treatments, monoclonal antibodies, CAR T cells, new understanding of the disease, and new technologies. No one can say today who will be the high-risk patient in 2025. A significant number of low and standard risk patients still early relapse from treatment, and maybe we miss the prognostic impact of clonal architectures, epigenetics, immune microenvironment, and many other things. And finally, do we need to define a risk at relapse? To try to answer this question, let me show this last slide. We now have been performing targeted next generation sequencing for every patient in our lab for 1 year now, a little more than 1 year. And you have the cycle plot of one patient at diagnosis. This is a hyperdiploid patient with trisomies 3, 5, 9, etc., classified as a good risk. And here in the inner circle, you can see his first relapse from an intensive treatment. And he acquired a deletion 17p on one allele and a TP53 mutation on the other allele. In other words, he became a very high-risk double-hit multiple myeloma. I'd like to thank you for your attention, and I'd like to address many thanks to all my colleagues from Toulouse and from the IFM, and in particular, Professor Hervé Aveloiseau. Thank you.
Thank you so much for inviting me here. Special thanks to Dr. Mahoti, Dr. Nagler, and Dr. Facon for this kind invitation for this wonderful meeting that has been going on now for a number of years. We are in a very difficult time right now, and that's why this broadcast, we have lots of dark clouds over us. We all hope that very soon a new day will come with brighter futures and brighter prospects. And this presentation online might end up becoming more routine just because we like it, then we have to do it like it is today. So these are my disclosures. Now, high-risk disease is defined in very many different ways. A standard definition used to be, or still partly is, patients dying within 2 years of their diagnosis. However, over time, this has changed a little bit because we have better treatments. We have 12 new drugs and lots of combinations, so they do better. And this definition has changed to some extent because of that. Now it is being stretched from 2 to 3 years. And or if you keep 2 years, the proportion of patients who fit that definition are smaller. So keeping that in mind, one can define high-risk disease. Now, clinically, we have reasonably different understanding of what that high-risk disease is. Patients with plasma cell leukemia at diagnosis clearly has a very poor prognosis. Patient with extramedullary, not bone-related, they have poor prognosis. Patient with CNS disease, high LDH, and patients who have obvious clinical sign of proliferative disease, the lesions, for example, increase within a short period of time, and circulating plasma cell as in plasma cell leukemia. So these are clinical definition of high-risk disease that we all know in our practice would carry a poor prognosis. And then there are therapeutic criteria which we also know, although not well defined, as those patients having a difficult disease. If you have received combination with proteasome inhibitor, with immunomodulatory drug like RVD or KRD combination, and patients have no response, we know those patients are not going to do well, or patients relapses very soon after transplant, or even while we're stopping treatment to collect stem cell, if patient starts progressing. Those are the patients who are poor prognosis. However, these are not well-defined criteria. This is what we know from our clinical experience. What is well-defined, genomically and otherwise, are very set criteria over a period of last 30 to 40 years. And the common genetic abnormalities as studied by FISH analysis that we identify are listed here, and their frequency also, which varies from as small as 5% to 50% with the various different types. However, there are certain features which carry poor prognosis. The most prominent one is 17p deletion, or along with mutation of 17p in 5 to 10% patient, and still carries poor prognosis despite having new drugs. 14-16 translocation, or to lesser extent 4-14 translocation, also carries a high risk category designation. And then newly evolving but also quite well accepted is amplification of 1q21 and deletion of 1p32. These are all changes that are considered to be having patients with high-risk disease. So this classification does not include the ISS stage, which includes two features, albumin and beta-2 microglobulin. So in the revised ISS staging system, which incorporates all the features, the fish-driven features, the ISS staging system, and also includes LDH, we know carries some weight into one. And in this regards, if patient has ISS stage 1, patient has normal cytogenetics and normal LDH, that belongs to RISS1. On the other hand, the revised ISS stage 3 are patients who have traditional ISS stage 3 with abnormal LDH and or abnormal FISH, and everybody else is in the middle. And these are very distinct groups, well defined, and have clear clinical relations in how these patients do. Now, if we then move on to seeing whether it can be applied uniformly, there's one more concept that is emerging, that even subclonality matters, in the sense that if the observed cytogenetics or FISH change is not in every myeloma cells, but only in the subgroup of cell, even then this carries poor prognosis. And the best example is deletion 17p. And the level that is identified is 60%. So if 60% patients carry 17p, as shown here, they have a very poor prognosis, both for PFS and OS. If it is less, the adverse feature is not as significant as it would be with 60% subclonality. So we also need to look for subclonality in some of these changes, and this will become an important component of our analysis as we move forward. More recently, what is identified is this concept of double hit myeloma, where combination of two of the features, two of the cytogenetics, genomic or clinical feature combined, can identify patients who are what we would call ultra high risk with a very short survival. And the two features are either biallelic dysfunction of p53. So patients could have 17p deletion on one allele and could have p53 mutation on other, making it biallelic change, or patient with ISS stage 3 and amplification of 1q, preferably 4 or more copies. And if you see here in the bottommost curve in blue, which was what our test data set was, that has the poor prognosis. These are the high-risk patients. Without these patients, double hit patients, have slightly better outcome. And similarly, in a validation set, an independent data set, the same group carries poor prognosis. So these are ultra high-risk patients who have really short, less than 2 years, between a year and a half overall survival and progression-free survival. And we have to keep this patient in mind as we devise our therapeutic plan. Then over last almost 15 to 20 years, expression profile has been used to predict outcome, and based on that, high-risk patients have a poor survival as determined by certain group of genes. This particular original was a 70 gene signature, which was used to define this high-risk group. Subsequently, the Hovan group has described a 92 gene signature, which is actually provides even a better result than 72 gene signature, and has been continued to be evaluated in various patient population. And between the 70 and 90 gene signatures, one continued to keep an eye on how the prognosis changes. An interesting component that I would want to bring out is that although we have all these various signatures with 70 gene and 92 gene, there's very little common between each of these signatures. There are, for example, there are just two genes which are common between the Hovan signature and the 70 genes UAMS signature. And that tells us that as far as expression is concerned, there is significant redundancy. And these expressions are based on the usual protein coding gene. And I'll come to the concept of non-coding genes, which may be playing an equally important role that in future we may have to incorporate. So defining high-risk disease by genetics, we consider a revised ISS staging system, look at the double-hit myeloma, look at the high-risk gene expression profile pattern. But then newer and more important component is the mutational burden, and how does that pan out? Now, we and others have done significant exome sequencing or genome sequencing and identified certain recurrent mutations in myeloma. NRAS, KRAS, P53, for example, are the three commonest, and they vary between 15 to 20 to 25% in all these data sets. And then BRAF and others are less frequent. Most of the others are less than 10%, and majority are actually less than 5%. There is nothing common, and none of these mutations by themselves individually carry poor prognosis. So although in certain studies you might find one of them having some impact, except for the P53, which especially in connection with deletion 17p may have some connections. However, what is important is shown in the right here, that not specific mutation, but the total number, frequency of mutation may carry significance and may identify high-risk patient populations. So patients who have high number of mutations have poor prognosis, as you see here in the light blue line. And that's what we have to keep in mind as we begin to redefine high-risk disease in these patient populations. Now, we have gone on to study the genome in greater depth by using whole genome sequencing and doing whole genome sequencing as much deeper level, between 60 to 80x. And we also studied various other genomic parameters here, which includes RNA sequencing, epigenomic profiling, and SNP, etc., in large number of patient population who are also had a uniform treatment, so we can predict the outcome much more significantly. And what we observe here is that when we look at the whole genome, the number of mutations are significantly high. It's around over 11,000 mutations are observed per patient, and there is a great variability as the rate shows here. And if you look at the indels, insertions and deletions, the small size ones, they're also quite frequent and variable from patient to patient. And if you look at the usual somatic mutations by indel, it's 738 per patient. And now if you look at various traditionally divided patients group by cytogenetics and FISH, the frequency of mutation is variable. 1416 is very high frequency of mutations, higher than any other. And few other categories, for example, 614 or 814 has relatively higher mutation, and deletion 17p definitely has slightly higher mutation burden. So higher mutation burden does suggest something that's more active in the disease, and we have to keep in mind. But when we look at the whole genome, where are the mutations? And what we find is that majority of the mutations, 95% and above, are in the non-coding area. And so what we have studied so far in the past, what I showed, with the coding region of the genome, which constitute around 2% of the genome, the non-coding region is where most of the mutations are. Some of them may be bystander, having no effect on the overall outcome or expression profile or something. However, some of these may have a significant impact on how the disease behaves and progresses. So looking at that, when we look at the top 20 hotspots in this area, there are only 4 which are in the coding region, and some of the well-known genes like BRAF or IRF4. But majority of the other mutations are in the non-coding region, and they have a significant impact on the outcome, as you see by the forest plot here. So we have to keep that in mind as we go along. The important component for today's discussion is that even at a whole genome level, the frequency of mutations matter. So patients who have high mutational rate, the group in the red here, have a significantly poor outcome compared to those who have fewer mutation. And so frequency of mutation, which suggests genomic instability, ability of genome to acquire many mutation, plays a very critical role in in the disease behavior in myeloma. And that might be our true high-risk group, patient population group. Now, what we have and others have gone on to study is what causes these mutations, and maybe that cause is of greater importance in deciding the high-risk disease. So earlier on, we had identified Niccolo Bolli from our group that there are 2 mutational signatures that are responsible for causing most of the mutations in myeloma. One is the age-related, but the other one is driven by ApoBec. And ApoBec is an enzyme activity which is important for some of the immunoglobulin-related changes, somatic hypermutation that we see. But its dysfunction may be important in myeloma. And Brian Walker subsequently confirmed this finding, suggesting its real importance. And if in his data, what he showed is that patients who have the highest ApoBec-related mutations, or you could say highest ApoBec activity, functionally looking at the mutation, has the most poor outcome compared to those who have less ApoBec activity. And you can see in the forest plot that for both PFS and OS, and the fourth percentile ApoBec activity has the worst outcome as far as the hazard ratio is concerned. Now, if we look at our own data, where we have also studied ApoBec, ApoBec here, Francisco Mora did. The point that was being confirmed in this was what Nicholas' data was, that ApoBec activity is present in large number of patients and carries the functional impact or the clinical impact. Then Dr. Walker, working with Dr. Morgan, have also identified similar finding that patients with predominant ApoBec signature, which is the cluster B here, you can see large number of ApoBec activity, they have higher number of mutation and carries poor prognosis. So having ApoBec signature has a poor prognosis, and it is an important endpoint to keep in mind. Now, when we analyze our data that I just showed, that in 200 patient deep whole genome sequencing, we identified actually 8 different signatures with critical impact in myeloma. These signatures are mainly related with 2 of the function. One is, of course, the age-related signature is important, is there, and it plays an important role. But then there are ApoBec-related signatures. There are 2 types of ApoBec, SBS13 and SBS2, as well as there are DNA repair-related signatures, the SPS8, and somatic hypermutation in lymphoid cells. And these signatures are quite important in how the disease behaves. There is also, as you see in this data slide, that within the various subgroups of myeloma, these mutations are either increase or decrease and may have some important connections. Now, most importantly, what we analyze using Mehmet Kemal Samur in our group has analyzed is that does these mutations start early or late? These functional aspects of all various signature, when are they active? If they're active at a clonal stage, that means that signature was active at an earlier stage of the disease, and this is the earlier stage of the disease. If you find the signature-related mutations at a subclonal level, then it's a late signature. And looking at this, you could see that, for example, DNA repair-related signature was much less in the beginning, but it goes up significantly as the disease progresses. And we believe these are the signatures that may play a greater role in disease outcome, drug resistance, becoming aggressive disease, etc. And so this is the one mechanism that we need to keep in mind as we look at high-risk disease in this patient population. The age-related signature here, which is high in the beginning, that may be related with earlier development of the disease, goes down subsequently in the disease process because it is at a subclonal level, it is much less frequent. And now if you look at various patient subgroup defined by cytogenetics and FISH, we find 3 different groups. We find, for example, that the ApoBec-related signature in majority of the patient, hyperdiploid myeloma patient, for example, start at certain level, but over time, over disease progression, it goes down. While in some of the high-risk disease, the ApoBec signature goes up during the intermediate phase of the disease, and then it goes down. On the other hand, the typical DNA repair-related signature goes significantly up in these high-risk disease populations. Also happens sometimes in 414 myeloma. So we have come up with a model of how the disease progresses. In one model, which is applicable generally, is that the initial disease process starts with an age-related and IGH hypermutation-related signatures. That goes down as the disease progresses, but then it is taken over by the DNA repair and SPS5 type of signature and mutations. And that's usually observed in a good risk myeloma patients. On the other hand, patients who have a high-risk disease, we find similar pattern. We find higher initial mutational rate, but very importantly, in the intermediate phase, there is an increased ApoBec signature. And this is something we have to keep in mind, how the disease is progressing and what we may need to target. So these and other studies that we have, Masud Samas in our group have performed, have identified various other processes that may be active in multiple myeloma. homologous recombination we have published many years ago, and its dysfunction may play a critical role in myeloma becoming aggressive and developing drug resistance. We have other molecular targets such as apex nuclease activity, pan nuclease activity. Of course, we talked about ApoBec, and various other DNA repair pathways such as NER and BER. So these are the processes that we need to study. We have already developed in vitro assay that we can quantitate some of this functional activity, and now we are in the process of correlating them with the outcome to find what I would call an ultimate measure of high-risk disease, where disease can progress very quickly. And then finally, very briefly in one line, how do we treat high-risk patients? That's not my talk for today. My focus is genomics, as we just discussed, but we have to keep in mind that in high-risk patients, we have to consider specific therapeutic intervention. Maybe we need to consider early use of novel agents. We now use 4-drug regimen that combines antibodies, and high-dose therapy also is an important component of the treatment here. We use the data to evaluate and establish long-term plan. Especially, we might continue multi-drug combinations for maintenance treatments, such as patients getting RVD for a long period of time. And we need to get MRD negativity to improve the outcome. So if you look at MRD negativity, data from Professor Avelova was that if you get MRD negativity, and this was IFM DFCI study where half the patient got transplanted, half the patient did not get transplanted. And if you look at patients who are high-risk disease, if they get MRD negativity, that's a red curve, or they are standard risk, which is blue curve. If they're MRD negative, their outcome is very similar, suggesting that if we get MRD negativity, we may be able to overcome a significant amount of high-risk characteristics imparted by cytogenetics and other features. So getting MRD negativity is important in these patient populations. And to evaluate high-risk disease, there is a concept being developed that at the time of diagnosis, we should look at the mutations, the copy number alternations, FISH, or other driven chromosomal rearrangements, and if required, gene expression signature to define low risk, high risk to decide the treatment. At the time of maintenance, usually we would look at the evaluation of the residual clone for MRD, positivity or negativity, and then apply the treatment accordingly. And when patients relapses, we have to reevaluate the mutational spectrum, the clonal evolution and signatures, because patients' myeloma change. Those who are standard risk to start with at some point can acquire high risk feature where we may have to modify our treatment. So change in the risk is an important component of a progressive disease, and that suggests that we need to measure this at multiple period of time. So with this, I would stop here. These are my colleagues in the lab who have done a lot of the work that I showed today. And then, of course, my colleague, Dr. Anderson, my bioinformatics colleague, and of course, my IFM collaborators, as well as my collaborators at the Sanger Institute. Thank you very much.
Hello, I am Evangelos Terpos, Professor of Hematology from the University of Athens School of Medicine in Greece. And during this COMy meeting, which is virtual, unfortunately, this year we are going to talk about the biology of bone disease in multiple myeloma. These are my disclosures. And as you all know, skeletal-related events are a serious problem for patients with multiple myeloma. This comes from a series of our patients that were presented in the last ASH meeting in 2019 in December, where you can see that the skeletal-related event observed at 44% at diagnosis, mainly fracture, as you can see here in 1/3 of the patients. 7% will need radiotherapy. This is much lower compared to previous publications in conventional chemotherapy, and this is because we have now very good anti-myeloma drugs. The need for surgery is approximately 6%, and the spinal cord compression is 5%. As you can see here, patients who have osteolytic lesions, 76% at diagnosis, have a more pronounced skeletal-related event compared with those who had no lytic lesions. Also, patients with abnormal MRI, as you can see here, have more often skeletal-related events compared to those with normal MRI. The most important finding of this publication is that during the first-line therapy, 6% of the patients develop skeletal-related events, even if they receive bortezomib-based, IMiD-based, or both bortezomib and IMiD-based regimen. So 6% of the patients during treatment will have a new skeletal-related event. And at the time of the first relapse, and this is the first publication that we have data for first relapse in patients who were treated upfront with novel agents, approximately 4.5% of the patients presented with a new fracture, and 12% will require local radiotherapy to the bone, ending up to 16.5% of the patients who are having a new skeletal-related event at the time of first relapse. The rate of skeletal-related event at first progression was much higher in patients who did not receive upfront bisphosphonate, and this is very important, while also during second-line therapy, another 12% of the patients will develop a new skeletal-related event. So in total, and this manuscript is now under preparation for submission, 20% of the patients developed at least one skeletal-related event during the course of the first and second-line therapy, and this was more common in those who presented with a skeletal-related event at diagnosis. As you can see, skeletal-related event, bone disease, is a common problem in multiple myeloma, and I think that it's crucial to see what's happening in the biology. But in order to understand the biology, we need to have some background regarding the normal biology of bone remodeling. So in our bones, we have the osteoclast that resorb the bone, we have the osteoblast that produce the new bone, but the most numerous cell into the bone is the osteocyte. Osteocytes are osteoblasts that have been entombed into the bone that have produced, and they are 90% of the cells of the bone. They link to each other with this microcanalischi. Every hour, as we stand up, as we walk or run, we have microcracks into our bones. And these microcracks disrupt the communication of the osteocytes. And these osteocytes go into apoptosis. And as they go into apoptosis, they release factors like the rank ligand and others that make the osteoclast to come to the area and resorb this old and cracked bone. At the same time, the osteoblasts come to the area in order to fill this hole. Let's see, for example, the microcracks here as they are in the real life in animal models. And you can see the osteocytes that link to each other with this microcanalischi. They look like the neurons, if we can say that. These are the healthy osteocytes. If we disrupt this communication, the osteocytes go into apoptosis. And you can see that as they have become apoptotic, they have numerous vacuoles into their surface that have molecules. We don't know all the molecules that these vacuoles include, but we do know two of them, the rank ligand and sclerostin. And you can see here the necrotic osteocyte. So as I previously mentioned, this microcrack makes the osteocyte to go into apoptosis. They produce a lot of rank ligand that is the most potent osteoclast activator. The osteoclasts come to the area, resorb this old bone, and at the same time, the osteoblasts come in order to produce new bone. When do the osteoblasts know that they have managed to fill this hole with new bone and it's time to stop? They know it because the osteocytes that underneath produce a molecule named sclerostin that is an osteoblast inhibitor. And the osteocytes feel the mechanical pressure. So the more bone that has been produced, the more mechanical pressure the osteocytes are feeling, and the more sclerostin they produce in order to make the osteoblast to stop working. So sclerostin is an inhibitor of the osteoblast and is a Wnt signaling inhibitor. Wnt signaling is one of the many pathways of the proliferation, differentiation, and activation of osteoblasts, but is one of the most important. You can see here that the Wnt protein binds to this co-receptor of the frizzled and the LRP5/6 protein, and enhances the overproduction of beta-catenin, that is an important transcriptional factor for the osteoblast. So the Wnt signaling, the canonical Wnt signaling that acts through the beta-catenin overproduction is very important for the osteoblast. We have two important molecules that inhibit this function. The first one is sclerostin, that I previously mentioned, that antagonizes the Wnt protein for the LRP5/6 part of the co-receptor of the Wnt protein, so it inhibits the canonical pathway. The second is DCOP proteins. DCOP proteins also use the LRP5/6 part of the co-receptor of the Wnt protein, and so antagonizes also the Wnt protein. So sclerostin and DCOP proteins inhibit the Wnt pathway, and they are inhibitors of the osteoblast function. But how about the osteoclast? You can see here in a real picture from electron microscope, color electron microscope. You can see the microcrack here. You can see the osteoclast that is activated, and the osteoclast is like jellyfish. You can see within the small foot of the feet of the osteoclast, the pH goes down to 2, and the osteoclast resorbs the bone. You can see the microcrack here, the hole that the osteoclast leaves behind in order the osteoblast to come and fill this hole with the new bone. And this happens all the time in our bones. The most potent osteoclast activator is the rank ligand that is produced, as I mentioned, mainly by the osteocyte, but rank ligand also produced by the T cells. That's why in every autoimmune disorders like Crohn's disease, like rheumatoid arthritis, systemic lupus, we have a lot of production of rank ligand by the T cells, and that's why we have osteoporosis. So the rank ligand, but locally at the bones, it is produced mainly by the osteocytes, binds to rank on the surface of perfusion osteoclast. Just to remind you that the perfusion osteoclasts are originated from a macrophage precursor cell that gives birth also to macrophages and dendritic cells. So at some point, the osteoclast is a hematopoietic cell. It has toll-like receptors in its surface, so it is a very interesting cell. So rank ligand binds to rank on the surface of perfusion osteoclast and enhances the proliferation of them to mature osteoclast and differentiation. And at the same time, the binding of rank ligand to rank on the surface of the mature osteoclast inhibit their apoptosis. If we don't have rank ligand, like in this rank ligand null mouse model, we have osteopetrosis because we don't have any bone resorption. As every important chemokine in nature, rank ligand has an inhibitor, and this inhibitor is osteoprotegerine. Osteoprotegerine is mainly produced by the osteoblast, binds to rank ligand, and inhibits everything that I previously mentioned. If we don't have osteoprotegerine, like this OPG null mouse model, you can see that we have very low bone volume, and as the osteoclast cannot be inhibited, in the OPG null mice, we have low bone volume and pathological fracture, as you can see here, and in the spine here. So the ratio of rank ligand to OPG drives the osteoclast activity not only in malignant diseases, but also in all disorders of the bone. So let's see what's happening in multiple myeloma. The adherence of myeloma cells to the bone marrow stromal cells leads to the overproduction of cytokines and chemokines with osteoclast activity. Very important are the MIPs, the macrophage inflammatory protein 1 alpha and 1 beta. Now MIP1 alpha is called CC ligand, CC motif ligand 3, CCO3, with the new nomenclature of the chemokines. These proteins are chemoattractants for the osteoclast precursors, so the myeloma cell attract the osteoclast precursors into their microenvironment in order to transform them to osteoclast. But in order to do that, they need rank ligand. So rank ligand is overproduced by the osteocytes in the myeloma microenvironment because the myeloma cell makes the osteocytes to go into apoptosis, as Nicola Giuliani showed very nicely some years before. And also, some myeloma cells produce low amounts of rank ligand. How about osteoprotegerine? The osteoprotegerine is downregulated in the myeloma microenvironment because the osteoblast cannot work very well. And furthermore, these OPG, these low OPG levels, are further reduced because the myeloma cell using the CD138 the very well-known cytokine-1, bind osteoprotegerin, internalize, and degrade it. So the ratio of rank ligand to osteoprotegerin is in favor of rank ligand, who have the activation of osteoclasts that resorb the bone, producing osteolytic lesions. And at the same time, as I previously mentioned, the osteoblasts are exhausted in the myeloma microenvironment because they are inhibited by molecules like DKK1 or soluble fusylated protein 2 that are produced directly by the myeloma cell, or by other molecules like sclerostin and activin A that are produced by the bone marrow stromal cells or the osteocytes. So we have the activation of osteoclasts, and this very complicated system gives back to the myeloma cell growth factors like interleukin-6, B-cell activating factor, insulin growth factor 1, and J-bril that makes the myeloma cell to grow into the area that the osteoclasts have produced. Keep in mind, rank ligands, sclerostin, activin A, and DKK1 that have now molecules that monoclonal antibodies that targets these molecules. We know from many years before, this one of my first publication, that the rank ligand to OPG is elevated in patients with multiple myeloma. Here are patients with no lytic lesions, 1 till 3 area of lytic lesions, more than 3 areas of lytic lesions or a fracture, and you can see how much more rank ligand to OPG is in the extended bone disease. And this rank ligand to OPG ratio, the enhanced ratio, correlates also with survival in the era of conventional chemo that was the treatment of myeloma back to 2000. And Karen van der Kerken with Peter Croucher showed at the same time in mice that inhibiting rank ligand using osteoprotegerin in this publication reduces the paraprotein level, reduces the myeloma cell survival, and enhances or increases the survival of the myeloma mouse. So you can see that inhibiting rank ligand will have the increase in survival in myeloma mice. Another inhibitor another enhancer of osteoclast function is the MIP1 alpha, the macrophage protein 1 alpha, or CCL3. And you can see here that the more CCL3 that is produced by the myeloma cells, and this comes from trephine biopsies, expression of CCL3 by the myeloma cell, the more they produce, the more bone disease we'll have. Again, here, no lytic lesions, 1 till 3 lytic lesions, more than 3 lytic lesions into a fracture, and the more MIP1 alpha we'll have in the serum of the patients, the poorer the survival of the patient. So we have another publication suggesting that an enhancer of osteoblast function correlates with poor survival of the patient. This is mainly because the osteoclasts support the myeloma cell growth. You can see here a culture of myeloma cells after 14 days in the absence of osteoclasts, and the same culture when the osteoclasts are into the environment. You can see how numerous myeloma cells will have. And here, the Samuel Jacobi from the Leeds Rock also showed that this is the proliferation of myeloma cells alone. If you add in the culture osteoclasts, you see that we have 5-fold increase in their proliferation. If we put osteoblasts into this culture, we have a reduction in this proliferation by 2 times. The osteoclasts and osteoblasts are antagonizing for the myeloma cell growth. The osteoclasts increase the myeloma cell growth, and the osteoblasts reduce it. That's why the myeloma cell reduces the osteoblast function, and DKK1 is one of the molecules that inhibit the osteoblast function. You can see that in myeloma patients will have more production of DKK1, and also stage 2 and 3 patients produce more DKK1 compared to the patients with stage 1 disease. And from animal models, we know that if we give a monoclonal antibody targeting DKK1, you can see that we'll see bone formation because we inhibit the inhibition of the osteoblast. You see here that we have the production of new bone in a mouse model that receives anti-DKK1 monoclonal antibody. And there's another publication that you can see that using a monoclonal antibody targeting DKK1, we see the healing of the osteolytic lesion in this myeloma mouse model. And also, using anti-DKK1 monoclonal antibodies, we have the increase in the osteoblast numbers and also in their function. You can see here the osteoblasts that are more in numbers and also in the production of alkaline phosphatase from the osteoblast if we use monoclonal antibodies targeting DKK1 in the myeloma mice. As I previously mentioned, the osteocyte is very important for the bone remodeling, and also myeloma cell makes the osteocyte to go into apoptosis, and because of that, they produce sclerostin. If we use a monoclonal antibody targeting sclerostin, you can see also that we see the healing of the lesions, because like the monoclonal antibody targeting DKK1, we have the inhibition of the osteoblast, inhibition that happens in multiple myeloma with this monoclonal antibody. In humans, we have seen that patients in multiple myeloma, even in the plateau phase or at relapse, have elevated levels of sclerostin in their serum, and also patients with fracture have more sclerostin compared with myeloma patients without fracture. And we published that some years before in International Journal of Cancer. And this is important because now we have monoclonal antibody targeting sclerostin. The name is romosozumab, has been licensed for the treatment of osteoporosis, and this was the publication in osteoporosis that you can see that compare to alendronate or teriparatide, the treatment for osteoporosis, bisphosphonate or parathormone, the teriparatide, the recombinant parathyroid hormone. You can see that using romosozumab, you have better increase in the bone mineral density of the lumbar spine and total hip. And we hope that we are going to have soon this drug for the treatment of myeloma also. Activin is another important molecule that is a TGF-beta superfamily member that enhances the osteoclast function and reduces the osteoblast function. We have seen that activin A is elevated in myeloma patients with advanced bone disease and correlates with markers of bone resorptions like the C-telopeptides of collagen type 1. And sotatercept is an antagonist of activin A that has been used in patients with beta thalassemia, for example, because it enhances also the hemoglobin production. And we have seen that if it is used with MPT in patients with multiple myeloma, we have an increase in the lumbar spine bone mineral density, and in the absence of bisphosphonate with the different doses of sotatercept, we have an increase in the bone mineral density of the lumbar spine in patients who received MPT. As I previously mentioned, activin A is a TGF-beta superfamily member. So if we use a monoclonal antibody targeting anti-TGF-beta, then we manage to have an increase in the bone formation. And you can see here the bone production in naive mouse model, but also in myeloma mouse model that have more production of new bone when this is given, and this is a publication that was reported last year, and it is good that we have now an anti-TGF-beta monoclonal antibody, and phase 1-2 studies in myeloma patients are going to start very soon. Periostin is another molecule, is known as osteoblast-specific factor that we have seen in patients with multiple myeloma, that is elevated in diagnosis, in the plateau phase, in that relapse, correlates with high LDH, with the stage of the disease, and with the presence of fracture. So it's another molecule that plays a role in the myeloma bone disease development. And semaphorin 4D is a new molecule that we know that is produced by the osteoclast and inhibit the osteoblast function. There was a Nature Medicine paper some years before, and we've shown in the Blood Cancer Journal last year that in the myeloma patients who have elevated level of semaphorin 4D, that this correlates with the stage of the disease, with the presence of osteolysis, with the bone resorption factors. So having all this in mind, the therapies that we have now for myeloma-related bone disease targets mainly the osteoclast, as the bisphosphonate and the monoclonal antibody targeting rank-like denosumab. But we expect that we are going to have new drugs like romosozumab that inhibits the sclerostin, or anti-TGF-beta monoclonal antibodies that target activin and other TGF superfamily members that are going to be in phase 1-2 studies very, very soon in multiple myeloma, in order to have more drugs knowing the biology of the disease for the treatment of multiple myeloma. And with that, I would like to thank you so much for your attention, and I'm happy to have questions. Thank you.
Welcome, everybody, and I'm sorry to be doing this under such circumstances. I hope you have all stayed well and healthy. My name is Keith Stewart. I'm from the Mayo Clinic in Arizona, and I'm here this morning to talk to you about the role of drug resistance in the use of immune-modulating drugs and cell mods. I'd like to start by, I'm sorry, we have a problem here. I'd like to start by presenting to you some facts about immune modulators that I think we sometimes forget. The first is that while the response rate to drugs such as lenalidomide and dexamethasone is very high in the newly diagnosed multiple myeloma setting, between 80 and 90%, I think it's important we remember one of the enigmas of use of these drugs, which is that the single agent response rate to thalidomide, when initially reported in people who are completely naive to immune modulating drugs, was only 25%. Subsequently, lenalidomide, studied by Paul Richardson and colleagues, showed that the lenalidomide response rate, presumably in patients who had received some thalidomide before, was only 24%. Similarly, when pomalidomide was introduced, again, the single agent response rate was variable between 25 and 50%, but overall approximately 30% as well. And finally, the latest entrant to the field, iberdomide, with dexamethasone having a 32% response rate. So I think it's important that we remember today, and I don't think I'll solve the question for you, but that 70% of patients are de novo refractory to these drugs. This is not to say that we haven't made progress. Obviously, patients who received iberdomide had been exposed to all three of the other agents, people who were exposed to lenalidomide, many of whom had already received thalidomide. So with each progressive improvement in the drugs, with their potency, we've continued to see responses. In fact, if we were living in an ideal world, we would be using pomalidomide today in our frontline setting and perhaps iberdomide in the future. The primary breakthrough, as you're all aware now, happened in 2010 when a group of scientists in Japan decided to tag thalidomide and pull out binding proteins in their search to look for the causes of embryonic damage. They found that one of the binding proteins was cereblon, a protein expressed in the brain with a LON protease, which if you're born with defects in this gene, lead to intellectual disability. They went on to show that cereblon, which was known to be an E3 ubiquitin ligase, was in complex with two other proteins, DDB1 and COL4A, and that together thalidomide bound this complex and changed its function in a way which resulted in limb defects in various models. We capitalized on this. We worked with Dr. Rob Orlowski at MD Anderson, who had generated a lenalidomide-resistant cell line, as shown in the top left here. We were very quick to show that, in fact, the resistant cell line had completely deleted cereblon, as shown on the bottom and in the top right, suggesting that, in fact, this was the mechanism of resistance in this cell line generated deliberately to be resistant to the drug. We next took a group of patients who had recently been treated at Mayo Clinic on a clinical trial of pomalidomide, either as a single agent or with dexamethasone. Fortunately, gene expression profile had been performed on these patients, and we were able to show that if your cereblon levels were basically normal for myeloma, the response rate was 33%. If they dropped below 90% of normal, it dropped to 19%. And if gene expression levels dropped below 80% of normal, there was no response. In other words, the complete absence of cereblon completely negated any function of this drug. In this study, we were also able to show that, not surprisingly then, that correlated with a change in progression-free and overall survival, as shown here, with quite a dramatic impact, in fact, such that patients with low cereblon levels had a median progression-free survival of only 9 months versus those with normal levels who had a median progression-free survival of 27 months. Thus, we knew at that point that, in fact, all of the immune modulators were, in fact, cereblon binding small molecules. We also noted the fact that as the drugs had become more potent, more specific, with stronger binding of cereblon, that the off-target effects had dropped, such that side effect profiles were better for lenalidomide versus thalidomide and pomalidomide versus lenalidomide, while the potency had increased. I've added to this slide the structure of iberdomide for your interest in the bottom right corner. Of course, we're still learning about the use of this drug in myeloma. The next landmark discovery came from a couple of groups out of Boston, predominantly, showing that these three drugs did something we didn't expect, which was they didn't inhibit the function of the cereblon complex, but rather they activated it in a way that the target client proteins, in this case the transcription factors Icarus and Aiolos, were ubiquitinated for targeted destruction by the proteasome. This had two immediate effects on intracellular function. The first was a downregulation of the targets of the transcription factors, IRF4 and subsequently MYC, resulting in cytotoxicity, and simultaneously the activation of the immune system with the upregulation of interleukin-2, hence the immune modulator term of the drugs, and the downregulation of tumor necrosis factor, which happened instantaneously along with downregulation of these tumor-promoting genes. An interesting corollary of this, if you think about it, is we've never quite understood why it takes so much drug in vitro to kill myeloma cells compared to other active compounds, suggesting that, in fact, T-cell activation may be quite important in the mechanism of action of these drugs moving forward. We went on to tag myeloma cells with a construct in which we deliberately linked Icarus to a green fluorescent protein. And what we've done here is line up myeloma cell lines for you from most resistant to most sensitive, and looked at the ability of lenalidomide to degrade Icarus. And you'll see that, in fact, there is a fairly strong trend, perhaps with the exception of one cell line in the middle on the right here, H929. Other than that, we see a very strong correlation with the ability to degrade Icarus and to have myeloma cytotoxicity, which is what we would expect. Subsequently, we show that this also segregates the three different compounds, such that the most effective degrader of Icarus is pomalidomide, followed by lenalidomide, and thalidomide being quite a lot weaker. One finding that was perhaps not surprising, but one that we still need to better understand, was that, of course, if you block the proteasome, as shown here, either with bortezomib or with carfilzomib, you block the function of lenalidomide. I'll show you this here. In the green arrows, we show the effect of having lenalidomide alone on suppressing Icarus function in the red line. And you'll see that when we add the proteasome inhibitors, we reach a point at which we almost completely abrogate this, both with bortezomib and carfilzomib. I will point out that the concentrations required to completely abrogate lenalidomide function are almost double that which are required to see cytotoxicity with bortezomib and carfilzomib, suggesting there is a therapeutic window, perhaps, during which you can still affect proteasome inhibition within a myeloma cell without necessarily completely switching off the function of lenalidomide. But this is a hypothesis rather than fact. So we turned our attention next to the human situation, the real-life situation of why, how important is cereblon in the function of these drugs. The first analysis of this was in a young woman from San Francisco who had unfortunately failed all available therapy, had a large extramedullary tumor on her chest wall, we biopsied and did whole genome, RNA, transcriptome, and germline sequencing to try and define what was causing her multi-drug resistant disease. And for the first time ever reported that cereblon was mutated in this young woman with a truncating mutation which completely abrogated its function. We then went on working with Mark Raab in Heidelberg and with Martin Kortum, now in Würzburg, Germany, to do a panel sequencing. The important thing to note about the panel sequencing is that the depth of sequencing is very high, 500 to 700x. And when we did this in patients who were quite refractory, we found 24% of patients had a mutation somewhere in the cereblon pathway that we consider to be pathogenic. Now, important to note that these were not clonal mutations. Something else that we don't fully understand today is that most of these mutations were subclonal, which is why we only found them when sequencing at great depth. I think the highest was around 50%, the lowest was around 8%, suggesting that even though these mutations emerge, they may not fully explain resistance, since the bulk population of tumor does not harbor these specific mutations. We were also able, with the help of our colleagues in Germany, to look back and find samples from 3 patients who we found a cereblon mutation to be present in, and we could find that earlier in their disease course, before they were exposed to immune modulating drugs, in the open white circles, there was no evidence of mutation, even using PCR, looking down to 1 in a million type of depth. So these are acquired mutations induced by drug, presumably resulting in refractory disease. One interesting side effect of these studies, this is from the group in Würzburg, Germany, that I've borrowed their slide, was to show that these mutations tend to cluster in 2 specific regions. First in cereblon on the left of the slide here, where you see the lenalidomide in yellow binding in the pocket there, and a lot of mutations clustered around that binding site. And a second group of mutations clustered around the binding between cereblon and DDB1. And in this particular lenalidomide-resistant patient had 7 different cereblon mutations representing different subclones of the disease. Santiago Barrio and Martin Pertum in Germany have gone on to extend this analysis and published last year on a much larger population of patients, 1,838 patients, in whom they found mutations in cereblon, Icarus, and COL4B, so IRF4 not included here, in only 6.2% of relapsed patients. However, I want to point out to you that this is done mostly on exome sequencing, where the depth of sequencing is only 30x, and thus only major dominant clonal mutations will be detected using this methodology. They were able, however, to show again that these mutations tended to cluster in 2 regions, either the thalidomide binding domain or at the DDB1 cereblon interface, and that these were significantly increased after treatment with a very significant p-value, as you might expect. We had noted earlier, perhaps even in our very first experiments with cell lines that I showed you a few minutes ago, that in fact, in cell lines at least, if you deliberately make them resistant to cereblon, it is usually a structural deletion, not a point mutation, that causes that phenomenon. And we had actually worked with some of our colleagues at Mayo Clinic, particularly George Vasmatis, who developed the technology, and Linda Bond, one of our molecular pathologists, to use whole genome mate pair sequencing as an alternative methodology of looking at structural change in myeloma. Now, it's not the topic of today's discussion, but I will tell you we have just recently published with those individuals that mate pair sequencing appears to be a much better way of determining genomic change in myeloma, as I'll show you in a second. Now, Linda took 70 samples sent into the molecular pathology lab at Mayo Clinic. Now, some of these were from Mayo. We know the provenance and we know their clinical history, but some of them come from outside laboratories where we don't have the facts about whether this was a late or an early stage myeloma. What we found in these 70 patients using whole genome sequencing was that 10% of them had a structural deletion in either Icarus or in Iolus in this case, or in cereblon. We know that 62% of these patients were relapsed, so we can estimate that the true incidence of this deletion is probably about a third higher, or 15%. We also know that in 4 of the cases that came from Mayo, all of the patients were relapsed and not newly diagnosed, again, supporting the fact that these are acquired and not de novo. This is an example of whole genome sequencing in a myeloma patient, and I will point out some of the advantages here that we can foretell with whole genome sequencing. Blue here represents, so black represents chromosome translocations, blue represents amplification, red represents deletion. You'll see that this is a fairly messed up genome in this particular patient. We could tell, however, that there is amplification of chromosome 11, 21, 17, part of chromosome 1, all of which suggests that this might be a hyperdiploid patient. We also, if you can look at chromosome 17, which is midway up on the right of the slide, you'll see this patient has a deletion of chromosome 17. And therefore, and also there is a MYC translocation into the kappa light chain, most of which would be missed using routine FISH. Importantly, given the discussion we're having today, if we focus our attention on chromosome 3, where the red arrow is, you will see that there is a deletion at the end of chromosome 3 in red. If we blow that up at the bottom of the screen, you'll see the deletion there with the green line representing where cereblon lies. And on the bottom right of your slide, you will see that that deletion completely takes out cereblon. Here's a second example. The genome here is quieter. You will see on the far right a translocation between the immunoglobulin heavy chain and MAF. You'll see at the top an amplification of chromosome 1. At the bottom, you'll see a deletion of chromosome 13. If you study chromosome 8, you will see a MYC rearrangement as well as a MYC translocation to the kappa light chain, all of which is foretold by whole genome structural variation as opposed to FISH. Again, if we look at cereblon at the end of chromosome 3, we see that in fact there are 2 deletions of the end of chromosome 3, one of which in the bottom right, again, completely abrogates the function of cereblon. And in this patient, I think there is no likelihood whatsoever that they would respond to any of the immune modulators had we this information available. We went on recently, Wenxiao Zhu and my lab went on to generate 4 pomalidomide and iberdomide sensitive and resistant cell line pairs. You'll see that the red line here represents the resistant clone, the blue line the sensitive clone in these 4 cell lines, and on the top here we see pomalidomide, and on the bottom we see iberdomide, suggesting iberdomide, like all of the other immune modulating drugs, of course targets cereblon as its primary function. And we next went on to look and try to determine why these cell lines have become resistant and sensitive. Not surprisingly, we found in 3 of the 4 of them, as shown here, the cereblon levels were either completely absent or much diminished. And we went on, although not shown on the slide, we went on to reintroduce cereblon into these resistant cell lines and show that they completely regained sensitivity to the drug. So if there were a way to replace cereblon or upregulate its expression, that would probably help us overcome resistance. There was one cell line, however, XG1, which behaved very differently. If here's my arrow here. If we look at what happened over here, we see that when you add drug to the resistant cell line, which is here, we see cereblon levels are normal, Icarus goes down, Aeolus goes down, and here we see there are 2 forms of IRF4, one of which goes down, but one of which does not. And consequently, MYC does not change. This, therefore, led us to do full whole genome sequencing on this cell line. We found that there was an interesting rearrangement of IRF4, which results in these short and long forms of the IRF4 protein here. And again, showing that one of the forms is diminished, but one is not. Thus, this whole pathway breaks down here. Interestingly enough, I don't know if it's a response to this or not, but we also found that in this particular cell line, there was extreme activation on the transcriptome of both interleukin-6 and STAT3, which we now postulate is another mechanism of resistance to these drugs. This went on to lead us to start to think about how we might overcome resistance in a case like this where cereblon is intact. We targeted IRF4 using a building off of a paper from 2016 in which the EP300 bromodomain inhibitor CBP30 was used. This indirectly results in downregulation of IRF4. And you'll see here that indeed, when you combine this E300 inhibitor with lenalidomide, we're able to overcome resistance quite effectively in the red bars here compared to lenalidomide alone. So this would be one particular methodology of inhibiting IRF4. We went on to look with Marta Casey and Leif Bergsegel as to whether this was true in an animal model. Marta and Leif have generated a human cereblon mouse. Now, for those of you not familiar with this story, it turns out that murine cereblon does not bind these drugs. That's any work that was done with unmanipulated mice with the immune modulators is probably incorrect. Subsequently, Leif and Marta introduced human cereblon into the mouse model. And you can see here in the top left of the screen that here is the wild-type mouse. Sorry, here's the wild-type mouse here. When you add pomalidomide, there's no impact on Icarus. Here's the human cereblon mouse. When you add pomalidomide now, we do see very nice downregulation of Icarus in response to the drug. So using this model, Leif and Marta have studied the effect of various compounds. Here is no treatment, dexamethasone, pomalidomide alone, showing some activity. The bromodomain inhibitor, E300 inhibitor here, pomalidomide and dexamethasone we see is very effective. Dexamethasone, the combination here that we know clinically works well together. And here is pomalidomide plus the bromodomain inhibitor, which is the most effective combination in this mouse model. So let me close there and tell you that what we've learned about this pathway to date are that mutations in the cereblon Icarus iolus IRF4 pathway are found in at least a third of relapsed patients, and this, I suspect, is quite a significant underestimate. We know it is rare in newly diagnosed multiple myeloma, that most patients have intact cereblon at that point. We've shown that by immunohistochemistry and other methodologies. We've also shown that the mutations we do see are probably passenger mutations at this point. I showed you earlier that these mutations are induced by exposure to the drugs, that really they are significantly underestimated by whole exome sequencing, which is most of the literature on the presence of mutations today. But however, when we do deep sequencing and we looked at structural change, about 34% and probably more, as I mentioned for numerous reasons earlier, I suspect this is closer to 40 or 50% of patients actually have mutation. It's not truly understood what the exact frequency is yet because it's been hard to document deep genomic sequencing and information in patients who are truly refractory and are only receiving single-agent drug. Since we don't use single-agent drugs very much anymore, it's been very hard to accumulate the type of samples we would need to do this analysis. Our work on structural variation and whole genome sequencing is very early. We've only looked at 70 patients, and they were a mix of newly diagnosed and relapsed patients, and we don't know whether they were refractory or not. I've totally neglected today to talk about the epigenome. I think Paulineri might cover that later, but work from Calgary with Nizar Bayless and Paulo, and from our own group in Mayo, Arizona with Leif and Marta, have clearly shown us that the epigenetic regulation of IRF4, amongst other genes, is likely, and MYC, for example, are likely very important in resistance, and I'll leave that for others to cover. And finally, we don't fully understand this issue of the discrepancy of the biology where proteasome inhibition blocks immune modulator function, and the discrepancy between that and the clinical outcomes, which are obviously very good when we combine these two types of agents together. So with that, I will close and thank you for your attention.
I'd like to start by thanking Professor Maury, Professor Nagler, and Professor Facon for this opportunity to present on the MRD as a tool to discontinue therapy in multiple myeloma. So the one question that is worth starting with is, why do we measure outcomes in multiple myeloma. Just think of MRD as just another different type of outcome, a shorter outcome that we're measuring. We do so to compare therapies. We do for prognostication, either for a group of patients or for individual patients. But what we don't do enough is to use short-term outcomes to modify treatment. Something that is routinely done in other hematologic malignancies, for example, acute leukemia or an aggressive lymphoma, where, for example, the lack of obtaining a complete response will trigger additional therapy, and conversely, the attainment of the deepest level response will often mean end of therapy. In myeloma, the progression or the lack of response often triggers change in therapy. I think few of us will continue a regimen that has not provided at least a partial response after a certain number of cycles, and definitely all of us would change therapy if there is progression. However, we very uncommonly change therapy in other circumstances. For example, we don't usually depart a regimen or add therapy in the newly diagnosed setting just because the patient did not achieve a CR. And that strategy of changing therapy because of not meeting a certain endpoint has really not been tested prospectively. Conversely, as we get better regimens that get more patients into a deeper response, and as the paradigm of myeloma, particularly the newly diagnosed setting, has become continuous therapy, we need to ask the opposite question. Is there a short-term endpoint that is adequate enough to enable discontinuation of therapy? So we know, let's look at first at traditional response criteria and see how that would serve us for a strategy of response-adapted therapy. As we learned from our colleagues from Spain, short-term response really translates into long-term benefit. For example, seen here in progression-free survival and overall survival. But in somewhat surprising way, we see there's a huge overlap of patients who get, for example, VGPR, a near CR, or a PR. They tend to really do very similar long-term, which is better than the patients who don't respond at all, but is not as good as the patients who have a response and are MRD negative. And here is even with MRD measure at the lower level sensitivity that we currently use. So when you look at this long term, we're really talking about three different groups of people. There's the super responders who do the very best. We have the responders, which are most patients, including the MRD positive CRs, the VGPRs, and the PRs. And then we have the non-responders. We are very accustomed to changing therapy to make a patient go from this third group to the second group. I don't think any of us will let a patient, continue on the same path if it's a non-responder. But we have not routinely, and certainly not in the setting of clinical trials either, test the approach of adapting therapy because the patient is on the first or the second group with either intensification or de-intensification of therapy. The idea of short-term MRD being a predictor of long-term PFS and OS was further demonstrated by a very robust initiative led by Dr. Munshi a few years ago, doing individual patient data, meta-analysis of several trials, and even with definition of MRD of 10 to -4, that effort showed that MRD among patients in CR is a strong predictor of both PFS and overall survival. So I don't think any of us would have any problem accepting MRD as one of the strongest prognostic, if not the strongest prognostic factor in myeloma. So far, we share data with 10 to minus 4, but as we know, depth of response does matter. And this was elegantly shown by the IFM 2009 study. When you look at burden of MRD at pre-maintenance, so after 3 cycles of RVD induction, transplant, and 2 cycles of RVD consolidation, or 8 cycles of RVD, the burden of residual disease very closely parallel the risk of progression after maintenance therapy. What is important to remember is those patients had a 1 year of maintenance and then discontinued therapy. So this data set really provides us the opportunity to observe the risk of progression in a setting where there is no further therapy according to different levels of response. If you are pessimistic, you would look at this data and say, well, even among the patients with the deepest level of response, they are still some ongoing progression. I like to look at that from the optimistic way to acknowledge that even after 3 years, the vast majority of the patients who had the deepest level of response have not had a progression, even though they are not on any maintenance therapy at this point. Perhaps most important, this data set also shows us that MRD post-therapy is not only a strong prognostic factor, but it might also abrogate other more traditional prognostic factors, for example, cytogenetics. So we can see here that long-term, the initially cytogenetically high-risk patients who go on to become MRD negative, although that happens less likely than it does among the standard risk patients. But if they achieve that short-term outcome, they tend to do almost as well as the standard risk patients who become MRD negative. And most of the badness of high-risk patients is carried on by the ones who remain MRD positive at the end of therapy. So MRD can become this dynamic risk assessment tool that might capture prognosis after therapy better than the initial risk classification. Our colleagues from the Spanish group further demonstrate that in a recent manuscript authored by Dr. Paiva, where they show that among the patients who become MRD negative, the risk of progression or death really doesn't change based on whether they start therapy as revised ISS 1, 2, or 3. Again, achievement of deep response abrogating the initial prognostic impact of cytogenetics. However, among the patients who are MRD positive, that's where you see the difference. Against the bad name of high-risk cytogenetic is really carried on by the patients who remain MRD positive. More intriguing is that they demonstrate the patients who become MRD negative after intensification have a similar prognosis that patients who become MRD negative already after induction. Again, if that confirms in the long term and in subsequent series, it becomes a very strong indicator that MRD negativity is a valuable endpoint on itself, not only a surrogate of sensitivity to therapy, but something that's worth pursuing even if additional therapy is needed. And that gives us great opportunities for de-escalation of therapy among the patients who become MRD negative, but also the opportunity of escalation of therapy to deploy new interventions on those patients who, despite optimal induction, often in transplant, remain MRD positive. There should be no reason to wait until those patients relapse to deploy novel therapies. And that's something that really needs to be studied prospectively. So knowing what we know now about MRD, I think we should start consider some new trial designs. For example, using different strategies for consolidation in patients who remain MRD positive after autologous, after optimal induction, because we know those patients are the ones who not do so well. So I think in the future it would be a fair game to take the default consolidation strategy, which is autologous transplant, and compare directly against new experimental immunotherapies, for example, CAR T-cell or bispecific antibodies in patients who remain MRD positive despite optimal, in this day and age, quadruple therapy. Another approach, more along the lines of de-escalation of therapy, is, we saw, for example, from the IFM 2009, that deferral of autologous transplant came at a penalty of shortening of progression-free survival. Well, as the induction therapy becomes far more effective than what we saw with 3 cycles of RVD, and now we have the opportunity to stratify patients by MRD, I think it's worth re-asking that question, but among the patients who have confirmed deep responses. So a possible design is to deploy longer induction, achieve an optimal response defined by MRD, and then in this subset of patients, randomized to immediate autologous transplant versus deferred autologous transplant. Another possible strategy is to use the notion of MRD response-adapted therapy, where patients have a path with several sequential blocks of treatment, in this case, induction, transplant, and blocks of consolidation. And the patients go from block to block of therapy up to a certain limit, as long as they do not meet 2 consecutive MRD negative reads. But if they do so at some point, either shortly after transplant or after one of those blocks of consolidations, they go into a treatment-free observation and MRD surveillance. I think those are all interesting designs that we should be pursuing in subsequent years. Professor Philippe Moreau was kind to share with us the design of the IFM 2020 trials. That is one that we certainly be very interested in the years to come, that takes that approach of using MRD to stratify patients to answer different questions. So use isatuximab, KRD, so a quadruple powerful induction regimen for 6 cycles. Observe here the longer induction that we have seen in most trials in transplant-eligible patients. And if the patients are MRD negative, they're asking the question, will 4 cycles of ISA-KRD replace transplant without penalty? So allowing for, again, transplant deferral in MRD-defined low-risk population. And conversely, patients who are MRD positive, does 2 transplant outperform 1 transplant plus a brief consolidation with ISA-KRD? The question of MRD enabling discontinuation of therapy is being answered more directly by the US SWOG 1803 trial. This is a post-transplant maintenance trial. The key question is lenalidomide versus lenalidomide and daratumumab for maintenance. But patients have an MRD assessment after 2 years, and if they are MRD negative at that point, they are randomized to continue therapy or to stop the assigned therapy. So this would directly answer the question, is there any penalty of stopping therapy on MRD negative patients after 2 years of maintenance? And whether the answer to that question depends on whether they are on lenalidomide or lenalidomide and daratumumab maintenance. Our group with the COMy consortium took on the task of doing MRD response adapt therapy by designing the master study. So this study uses daratumumab, KRD as the backbone, has 4 cycles of induction therapy. The patients have an MRD read at that point. They go on to receive autologous transplant, followed again by MRD read by next generation sequencing. The patients who happen to have the first and the second MRD negative, they go on to a treatment-free observation and MRD surveillance, where MRD is looked at after 6 and 18 months of observation. Otherwise, the patients go on to receive DARA-KRD for up to 2 blocks of 4 cycles each, with MRD read at each step. And again, once you have 2 consecutive MRD negative results, the patients go on treatment-free observation, MRD surveillance. Otherwise, if they end the whole treatment and don't meet that criteria, they go on standard of care maintenance treatment. These are the preliminary results, and we're now a little bit over 100 patients, which is a slight update from what we presented at ASH last year. And will not surprise you that this is a very powerful regimen, and we reach 82% of the patients become MRD negative to 10 to minus 5. Because NGS allow us to look at 10 to minus 6, we can also provide that data, and 63% of the patients are getting MRD negative to 10 to minus 6. There is some, not surprising, better results for standard risk patients than there is for high risk patients. But what is interesting is that difference doesn't seem to be that much, and it will be interesting to see as we continue to accrue to this trial with an enrichment for high risk patients, whether how those numbers are going to compare long term. Perhaps the most interesting aspect, however, is to see how often patients achieve those milestones. And the experience so far, this is an ongoing study, so the information here is quite fluid. But we see that about 1/3 of the patients are, super responders, and they are able to discontinue therapy after autologous transplant. So for those patients, therapy is very short, 4 cycles of induction, autologous transplant, and treatment is complete. The most common scenario is patients go on to receive 4 cycles of DARA-KRD, and then a consolidation, and then they go to treatment-free at this point. So the first MRD negative read is after transplant. This is the second they go off therapy. There are a few patients who are kind of slow responders who reach that milestone after 8 cycles of consolidation. And we think it's going to be, at the end of the day, only about 15% of the patients who never meet those milestones and go on land maintenance. The important thing is, we have now 48 patients that have reached this treatment-free observation and MRD surveillance. Follow-up is still short, is anywhere from 0 to 16.9 months. So the patient who is further along is soon reaching the 18-month MRD read. But among those 48 patients, we haven't had any patient progress, and we have not had even any patient have resurgence of MRD at the 6-month follow-up. So it will be very interesting to see as we get more patients in this phase and we gain longer follow-up, particularly with the MRD at 18 months to see what the rate of MRD resurgence is on those patients. Of course, we hope it's going to be very low. So what have we learned from the master study? One thing is that MRD-guided therapy is feasible in 95% of the patients using NGS. There's only a few patients that don't have a trackable sequence on the regional bone marrow that enables this test to be performed. With the good news is with quadruple therapy and autologous transplant, most patients will reach MRD negative CR. We choose to enable treatment-free observation with confirmed MRD less than 10 to minus 5. And I really cautions against any strategies that make such a radical challenge of paradigm based on one single sample, one single assay. As I said before, because it's NGS, and although we are operating with 10 to minus 5 for most of, to essentially, because that's the IMWG definition of MRD negative threshold, and also to better align with regulatory authorities, we can look at results of 10 to minus 6. In most patients, as you saw, they have a confirmed MRD to 10 to minus 5. That MRD burden would also be less than 10 to minus 6. And one thing that is not easily measurable, but it's very rewarding, is the patients do value tremendously be going off therapy. When we launched this study, one of our biggest concerns is that we're going to be resistance among patients on discontinued therapy. 100 patients into it, there has not been a single case where a patient complained about coming off therapy. The patients understand value, and they feel very reassured by the fact that you can provide MRD surveillance. So I think we're going to hopefully, as in the future, see more trials that include treatment-free observation and MRD surveillance. Maybe one day we'll even call this, TREFONs, for like we need another abbreviation in myeloma. And that should really be pursued as an alternative to the paradigm of continuous therapy. I think it should only be pursued in patients with confirmed MRD negativity. MRD surveillance should be used to mitigate the risk of omitting maintenance, allowing us to respond to MRD resurgence before we have biochemical and certainly before we have clinical relapse. That risk of omitting maintenance needs to be balanced against, quality of life. I think those studies really need to try to capture a quality of life that, at bedside is very different if patients are or are not on maintenance therapy. Cost needs to be a factor as we intensify the initial therapy, trying to get a deep and long-lasting, and hopefully in many cases permanent response. Those regimens becomes more and more costly, so they're not going to be acceptable unless you have a cost-containing measure, and that omitting a maintenance certainly helps to make that case. And definitely the toxicity of continuous therapy versus observation, including hematological toxicity, infection, and the risk of second malignancy. Perhaps the least emphasized of all the advantages is one that is, I think, scientifically most interesting. I think if you look at myeloma in its history, we have for a long time had therapies that did not provide a deep response. So the patients are living with a high disease burden for most of their remaining lives, or we have a paradigm of continuous therapy. So we have not really had a setting where we can observe in an undisturbed manner a situation where the patient is, disease-free by all measurable accounts and is not on continuous therapy. So that it would allow us to observe immune reconstitution, and I'm particularly interested in the interplay between immune reconstitution and molecular recurrence. And with that, I'd like to complete by, again, thanks to the organizing committee, and looking forward to meet you all, see you all in person in Nice in October. Thank you so much.